Hey everyone, my name is Xander, and this is my local expert project on Methyl-Seq. Methyl-Seq is also known as bisulfite sequencing, and it's a way to study epigenetic effects caused by DNA methylation. We're looking for CpG sites or islands, so a CpG site is a cytosine followed by a guanine, and a CpG island would be a lot of these in close proximity. You want to look at cytosine methylation because methylated cytosines will actually stop RNA polymerase from binding, so it can inhibit gene expression. In class, we've talked a lot about mutation-based changes, and that can include CRISPR going in, changing a nucleotide, knocking out a gene. Maybe you have a virus that invades a host and deposits its genetic material into the host's genetic or genome. And maybe you have your own cells that are repairing DNA damage and using non-homologous end joining to do it, creating indels. All of those cases can cause mutations in your DNA, can create different amino acids, which create different proteins, which can create different phenotypes in organisms. Epigenetics wants to look at the case where, well, what if I have different organisms with the same DNA, but they have different phenotypes? So no mutations, different phenotypes. What would that look like? Might look like this. So this is from Loki, season 1, and Marvel explains this as different variants of Loki throughout time. But in my case, what if these are all Loki? They all have the same DNA. How do they have different phenotypes? Why do they look different? Why does this guy look like Tom Hiddleston? Why does this Loki look like Thor? And why does this Loki look like Jim from The Office? They all might look different because they may be expressing different proteins due to DNA methylation. So that's why we would look at this. Okay, so a methylated cytosine is gonna have a methyl group on it. So your normal cytosine's here, and then methyl cytosine is here with your CH3 methyl group. And in the case where you're repressing gene expression, you are going to have a lot of DNA methyltransferases that bind and then methylate your CpG sites. And then those are going to be recruited by histone proteins, so H3K9, H3K36. They're going to be recruiting DNMTs, methylating your DNA, and then preventing RNA polymerase from binding. You'll also have other epigenetic effects going on, so sometimes these DNMTs will come in here and remove this histone acetylation group. And when these get removed, your histone proteins actually become physically more tightly packed together, which just restricts the area that RNA polymerase can bind. Now, during active transcription, you'll have different histone proteins. So these H3K4 proteins actually prevent DNA methyltransferase from binding, and that allows gene expression to continue. You'll also have these proteins, these TET proteins, which will remove a methyl group altogether from your cytosine and create hydroxy methylated cytosine, but it removes the methyl group, so allows RNA polymerase to bind. And you'll see this in cancer. So in normal cells, you'll be in a binary state. So either you have H3K4, which prevent DNMTs, allows gene expression, or you have H3K36 or other proteins which recruit DNMTs, methylate your genome, and then inhibit gene expression. Cancer during carcinogenesis kind of throws all that out the window and says, okay, well, you could have H3K4, but you might just lose all the other ones. And so now there's nothing stopping recruitment of DNMT. You might have H3K4, and then you might have substitutions. So now you have all these H3K36s coming in here. You might have other histone markers that come in that inhibit gene expression. Or you might just lose everything, and then there's nothing stopping cytosine methylation. And this could be important because in cases where you're, say for example, expressing a tumor suppressor gene, during cancer formation, now that same tumor suppressor gene may be inhibited because it's being methylated. Methyl-Seq also ties back in with what we talked about in class during RNA-seq. So the whole point of RNA-seq was to quantify whether you have up or down regulation of your gene. Say, do I have more expression? Do I have less expression? Methyl-Seq might help you answer the why. So you might have less expression of your genome. Why is that? Well, maybe that gene is being methylated. So this kind of help. It's just another piece of the puzzle. It helps fill in the picture. Workflow and limitations. Thankfully, the workflow here for Methyl-Seq is similar to what we've done for normal PCR with one additional step. So you'll have your double-stranded DNA, and some of these cytosines are methylated. You'll denature your DNA. You'll treat them with bisulfite, and bisulfite you can just buy in a kit. So, there's a bunch of companies that offer this. And bisulfite, what it does is it converts your unmethylated cytosines, so these, into uracils. And then during PCR amplification, these uracils get amplified as thymine. And then you can actually map these back. So I have my thymines here. We look back at our original genome. These were all cytosines. So then I could say, okay, well, I have thymine here instead of cytosine. So I know that this was unmethylated. And then the cytosines that stayed cytosines, I can say, okay, well, these must have been methylated. In this study, they were doing direct PCR sequencing or subcloning. So when you actually get your PCR product, you could just clone the PCR product as it is, and this will get you sort of like an average status of methylation across what you're looking at. Or you can break it up into pieces and then put it into vectors and clone that, and that kind of gives you more granularity because now you're looking at multiple versions of the short reads, and you can kind of add that up and get more coverage. So it just depends what you're doing. Some of the limitations, one of them is this hydroxymethylcytosine group that I talked about earlier. So it's good that TET gets rid of it. It turns the methyl group into a hydroxyl group. It gets rid of the methylation, allows RNA polymerase to bind. But it's bad for us because bisulfite doesn't convert this cytosine into uracil. And we want it to. We want it to convert all non-methylated cytosines into uracil, so this guy and this guy. But the hydroxyl group actually stops that from happening. So that can be a little tricky when you're doing bisulfite conversion. Thankfully, there are protocols that you can follow. There's different kits, different ways to take hydroxymethylcytosines into account. I won't be getting into that here, but there are things you can do. The other problem with bisulfite is it's hard to get complete conversion, and you have a lot of DNA degradation. So from the study, maximum conversion rates of cytosines occurred at 55 C for 4 to 18 hours and 95 C for 1 hour. But under those conditions, 84 to 96% of the DNA is degraded. So you want to expose your DNA to bisulfite long enough so that you have full conversion of your unmethylated cytosines. But you don't want to do it too long because you don't want to degrade all your DNA. You want to have enough to actually do PCR amplification with. So this is kind of a balancing act, and that's kind of what makes this method hard. You want to convert enough cytosine, but you don't want to leave it long enough to where everything is just gone. Some of the controls. So some of the controls you'll see are some controls that we have in a lot of the methods that we've looked at. You'll have a positive control and a negative control. You'll have fully methylated cytosine where all your cytosines are methylated, and when you undergo bisulfite conversion, you'll see no changes here, or hopefully no changes. And then you also have a negative control where you have all your cytosines are unmethylated, and so now you see all these thymines in place where they should be cytosines as indicated by the star. And then just one other thing to keep note of here is DNA methylation predominantly occurs on cytosines followed by guanine residues. So you might have a cytosine and then a thymine, or a cytosine and then an adenine, and those are just not as commonly methylated as CpG sites. Just good to keep that in mind. It's good to understand your reference genome and what your DNA is supposed to look like. And then these are Sanger sequencing reads. So, for example, your methylated control here, you'll have cytosines, and then in the unmethylated cytosines, you'll have these thymines. And then your other thing that you're looking at. So here's maternal DNA and fetal DNA. You'll have cytosines here because these groups are actually methylated. So you can do bisulfite sequencing with Sanger sequencing or whole genome bisulfite sequencing. sorry, you can do Methyl-Seq with either of these two methods. And Sanger sequencing, the good thing about it is it's cost effective, I guess, relatively. I mean, I think it's expensive looking at the prices, but it's at anywhere from 50 to 200,000, whereas NGS methods can be 100,000 to a million. So I guess relatively speaking, it's inexpensive. It's also quick turnaround, so you can get the results back in a week. And then visually readable is probably the biggest thing. So as humans, if you get all this data, all this matrix data, I mean, you're not going to know what to do with that. But if you get something like this, where you're looking at the colors and I see, okay, well, this is clearly guanine, guanine, adenine, you can make sense of this. And then on a Sanger sequencing read, okay, well, it's telling me this was a cytosine, or this was unmethylated because it changed from cytosine to thymine. It was unmethylated, and so the bisulfite actually converted this successfully. So you could tell where your methyl groups are here. So that's probably the biggest advantage to Sanger sequencing. Whole genome bisulfite sequencing, the best thing about that is the barcoding. You can handle millions of DNA molecules concurrently, map that back to a reference genome. You get a huge data yield, so you can have 50, 100 times coverage where you're really super sensitive to mutations, and you can see what exactly is going on. And then here's just a comparison. So between Sanger sequencing and Illumina, that's how you would do this. So you could use sequencing by synthesis. The one step it doesn't show here is your bisulfite conversion. But in both cases, you'll get a readout of your DNA sequence, and you could check back to your reference genome whether that's supposed to be methylated or unmethylated. The one big downside here is that both of these techniques expose the DNA to bisulfite, which degrades it. So thankfully, there's like a third generation sequencing approach called SMART sequencing, which is single molecule real-time sequencing. And for this method, you don't need chemicals, you don't need enzymatic conversions. You're just looking at the kinetics of the DNA polymerase. So what do I mean by that? You're looking at IPD, which is the interpulse distance, or the time between additions of the nucleotides. So let's say DNA polymerase adds this thymine. And then how long does it take before it adds this adenine? That's the interpulse distance. And then maybe you've added a nucleotide, and now how long do I see that fluorescence? And that's the pulse width. And it's also looking at sequence context. So here I have TAAC. And maybe TGCC is different. And so the context, the order of the nucleotides matters. And it combines all of that data into a matrix and then uses that data to make a methylation call. So the way this works is you'll have your DNA molecules here, you'll circularize them with these hairpin adapters. You're going to ligate these together, add in a DNA polymerase, and then DNA polymerase is going to create strands of DNA. And when it gets to these strands, so you have this blue strand over here, you have your red strand over here. When it gets to these strands, that's when you're looking at these kinetic values, so interpulse distance and pulse width. And because it's circular, you can do this a number of times to increase your accuracy. So for example, here on this red strand, you'll have GATA, CGTTA, and you're looking at this measurement window, which is about 10 nucleotides or so in either direction. Here, we're just showing a few. So you have ATAC, where your middle C here is your methylated cytosine or your cytosine of interest, is what you're looking at. And you'll go over here to A. It's 3 to the left of your cytosine. And you'll try to say, okay, well, 1, 2, 1, 2, 3, and then you'll kind of fill that in for the adenine. So what's the interpulse distance? What's the pulse width here? And you'll add that to the matrix. And then you're doing that for all of the nucleotides, again, within the measurement window on either side of the cytosine that you're looking at. And you're doing it for both strands. So this strand is the red strand, and then you're doing it for the blue strand. Then you're putting all that data together, and you're throwing it through a neural net. You're using a machine learning algorithm, you're using a neural net, and then you're coming out with a methylation score. So is it methylated? Is it not methylated? And the cool thing about smart sequencing right now is it's 99% correlated to bisulfite sequencing. So bisulfite sequencing is the gold standard. And what this means is that when smart sequencing says, okay, the cytosine is methylated or the cytosine is unmethylated, 99% of the time it's getting the call the same as what bisulfite conversion or bisulfite sequencing would say that that cytosine is. The downside here is that the sensitivity is only 90%, so it's only catching about 90% of the methylated cytosines. And the specificity is 94%. Sorry, the specificity is only 94%. So that's saying it's only catching about 94% of the unmethylated cytosines, whereas like Illumina sequencing, for example, is 98 to 99% accurate. So there's still a ways to go with this before it's as accurate as something like Illumina, but it's great because it uses machine learning and it doesn't destroy your DNA. Okay, well, I hope you enjoyed, and thanks for watching.